My name is Cash, and I'm working on epigenetics, and that work is going on in Benaras Hindu University, and I'm looking at the developmental aspect of mammalian systems, and one of them is like embryonic gonad. So I'm working on mouse basically. So it's mammalian system. So we are looking at the developmental aspect of this gonadal systems, and the most important aspect is we are looking at the gene regulation aspect of this one, like how genes are regulating a particular developmental pathway. So that we can understand that developmental pathway at molecular level, like what kind of molecular interactions are there, what kind of regulatory elements are there, which actually helps in the development of this particular organ. So if this organ is developing, that is gonad on which I am working, what kind of molecular interactions are there, how genes are playing a role within the system. And the most important aspect is we are also looking at the epigenetic level of regulation. So in case of epigenetics, what happens is like, in general we know there is a DNA in each and every cell. That DNA actually has regulatory elements which are actually responsible for expression of a particular gene. Expression means transcription and then for the protein. So if it is so, that gene will be able to perform some functions. But in case of epigenetic regulation, even though element of the DNA element is same, but regulatory elements can be different on the same DNA stretch. So these are kind of regulation in which sequences of the DNA will not change, but some other modifications will also be there on the DNA or either histone or some small RNAs, so that they can regulate the particular gene. So this is like, for example, as I'm working on gonadal system, say how gonads are developing, like testis formation and ovary formation, how these organs are actually forming during embryogenesis part. So if embryos are developing, so every organ is developing. So we are looking at a particular organ which is known as embryonic gonad. So gonad is developing from a bipotential organ. In that bipotential organ has potential to develop either into the testis or either into the ovary. So in case of this bipotential gonad development, common genes are there that is present in both male and female. So if these genes are there, they should express in both, or they should not have differential expression. But in case of this organ, even though genes are present in both, as I'm telling you, epigenetic regulation has no sequence bias. So in this case, what happens? Genes are present in both male and female as well, but these genes are getting switched on either in male or either in female. So this switching on and switching off of the genes actually regulate the formation of either testis or either ovaries. So these formation of differential organ depends on the differential expression of these genes, and that is not present on DNA elements. Why? Because sequence is same. I'm doing epigenetic part of it. For example, how epigenetic modifications are different in males and females during the development actually. So if there's any difference in epigenetic level, there can be possibility to regulate a particular gene either in male or either in female, so that this organ formation will help in that direction. The best aspect of being a scientist, like you can serve humankind, or you can serve a lot of new things to the society which is not known yet actually. And the most important aspect is like you should be always true to your work. Like for example, whatever you are doing, you cannot manipulate that kind of situations, because if you are manipulating, next generation will get the similar kind of things, and they will start working on your own work again. So they will again getting wrong ideas and all this. So never manipulate your data, never manipulate your thoughts. Present whatever you are doing as such. This is what I want.